And so anyway, let's talk about some of this stuff. So just a quick reminder, test number 3 will be 1 week from today, and it'll cover topics 3, 4, and 5, which some of this material might not be covered till Monday. So what I will do is on Monday, I'll make you a study list of the important concepts. So, but all those are relatively light, short-ish kind of units. Topic 3 is maybe the longest one. So.
Hi, Blaine.
Hello.
It's Brandon here.
My computer is not advancing to the next slide. Oh, there we go. Good to see you, Brandon.
Yeah, I watched the lecture yesterday, and I'm aware of the test in the 3rd, and and Debbie and I were work in progress on the big project, the water treatment plant project.
Okay. Yeah, we'll talk about that when we get there, I guess. So like I said, for the test, what I will do is on Monday make a list of kind of the important concepts to study for that, just so that there's no confusion about that. But same format as usual. There will be some multiple choice, some short answer, some calculations, those kind of things. So last day we started on topic 4, and we were talking about calibration and why it's done, and you've all had a little bit of experience in the lab calibrating at least pH meters and conductivity meters. And most instruments, there's kind of a similar type of technique. We're going to get a chance to calibrate turbidity meters in the lab as well. And you'll see the turbidity meters, there's not a probe, but it's an instrument, and there's some standards that come with the instrument. And it's kind of a similar procedure where the instrument will ask you for, at a certain point, putting in a certain standard, and the internal electronics will measure it and determine exactly how to put that standard and to make baselines. So we had finished off last day talking about, I had introduced this concept of spectroscopy, and so spectroscopy is kind of like an eyeball looking at something and seeing how dark or how light it is. And we're doing this a little bit more quantitatively. So what you're looking at here is kind of the internal guts of a spectrophotometer is what they're called, the instruments. And you can see basically you have a light source over here. There's a system in the between the light source and the sample that is going to select a wavelength. So, you know, a light source is going to have all the colors of the rainbow, and so there's usually some sort of system, a grating or a prism. There's a few different methods to do so, and usually select a specific wavelength. So let's say 525 nanometers or something like that. Then you have your sample. So you can see in this case, what I have is a pinkish, purplish sample, and then on the far end, there's some sort of detector, and the detector can measure how much light has actually gone through that sample. So this procedure or this method is called measuring absorbance. So how much light of that particular wavelength is absorbed. And so, like I said, this is kind of something that we naturally do with our eyes, but we're not really very quantitative at it. We can say that the Kool-Aid is a dark purple or a light purple, but you know, our eyes can't really give a good number for this. So that's why we have these instruments to do so. So you're probably familiar with a lot of colored things, since we kind of live our world in colors. There's all sorts of pigments out there. A pigment that you've probably heard of is, of course, chlorophyll. And what color is chlorophyll?
Green.
Green, of course it's green, yeah. So chlorophyll, there's actually 3 or 4 different chlorophylls. The 2 main ones found in plants are called A and B, and they absorb in the blue and the reddish regions of the spectrum. So you can see that's what's shown here in this little graph. You've got blue over here, and we've got red over here, and so that leaves the green in the middle. And so since they absorb the other colors, they show up as green. Mm-hmm.
Blaine, that's a fun fact about chlorophyll. Like, have you ever experienced that? Have you ever accidentally scraped your knee or pants on like grass and there's like green stuff on it?
Yeah.
That's the chlorophyll.
Yeah, I remember my grandparents' 50th anniversary. I must have been about, I don't know, 10 years old, and my mom bought me a brand new pair of dress pants, and I totally did that to them. So yeah, so absorbance are the colors that are not transmitted, but they're literally absorbed by the compounds. I just want to point out a couple of things about chlorophyll A and B. Something that you might talk about next year in organic chemistry is if you look at the structures, so notice there's like a double bond here, and then we got a single bond, and then a double bond, and then a single bond, and a double bond. So anytime you see those kind of systems, that's called a conjugated double bond system. So you have single double kind of thing, right? And what that means, if you remember some of your Chem 101, you may remember those pi electrons are kind of orbiting above and below the nuclei. And those pi electrons, when you have conjugated systems, can actually travel throughout that entire system. And what that means in terms of energy levels is that's really good at absorbing light in the visible and ultraviolet range. And so a lot of pigments that have colors have these kind of systems. Not always, but very, very common. So here's another pigment. This is beta-carotene. This is what is making carrots kind of reddish, orangish color. There's a few different carotenes out there, by the way. You may be familiar with other reddish vegetables like tomatoes. So they're not quite as orange, but you know, the actual color often has a little bit to do with not just the one pigment, but often there's other things making it colored. So you can see in this case here, the absorbance is mostly in the blue and green range, which leaves the red and orange and yellowy kind of colors left over. So notice that this particular pigment also absorbs over here in the ultraviolet range. So like I said, when you see these conjugated systems, sometimes there's absorbance in the UV range as well. So here's another compound. I'm not sure what the name of this is. Oh, it's up there. Cis-trans 1,3-cyclooctadiene. This is not a colored substance, but it does absorb in the UV part of the spectrum. So some of these things can be, I guess if you think about it, seen by detectors, just not by the human eye. One thing I've always wondered, and I don't really know the answer to this, is whether, and I'm assuming this is true, that certain animals must have slightly different ranges in the spectrum where they can see. I'm thinking like insects and things like that. And I wonder whether they can see in the ultraviolet range. So what are some applications of these things, right? We have these nice colored compounds. And so like I said, you know, when they're colored, it's obvious that it's there. And so one of the biggest applications of this kind of technique is really just measuring concentration, because we can do this quantitatively. So you can actually determine the concentration of something by looking at how much color is there. Now, like I said, we can't do this with our eyes. They're not very good at measuring, but the instrument can do so. So you can see here are some things that we could measure the colors of and how much absorbance is done through the instrument. So this leads to something called the Beer-Lambert Law. These are two guys, and I think they actually, I don't think they actually worked together. I think they were some chemists or physicists that actually kind of came up with this whole thing independently. And but it was named after them. Sometimes you just call it the Beer's Law as well. I've seen that as well. And this is something we're going to make use of in this week's lab. And you can see this is the formula here. So what we have is absorbance, and we have concentration. Now I don't know why they always use a little c, because in my mind concentration is usually a big C. But you can see really that's showing that absorbance and concentration are directly proportional to one another. The other two units there is a constant, and that constant is going to be specific for a specific pigment. So every pigment and temperature and solvent might have a different epsilon. And L is actually the length of the solution or the thickness of the test tube. So the little test tubes we use are called cuvettes. They're kind of little square things. See if I can draw a 3D square. Well, not too bad. So the standard length, by the way, on all these cuvettes is almost always 1 centimeter. So if you can standardize the width of the solution, and then basically what you have is a constant here in the middle. So really, this is kind of the big thing to know, is that absorbance and concentration are directly proportional, and that we have to figure out exactly at what rate is often actually determined in the lab. So, hold on a second. Yeah, okay, I'll come back to that calibration curve in a second, because I want to talk about what you're doing in today's lab. So, you know, this is just saying, oh, by the way, you know, and relating to this week's lab, some things are colorless, and we can't measure them by spectroscopy. But kind of like titrations, what you can do is you can add some sort of colored agent. And that colored agent will react with the compound you're trying to measure and produce a color. So this is what we're doing in this week's lab. We're going to be measuring iron. And iron, yes, it has a little bit of a color, but at the concentrations, mostly what we're measuring at it is so small. So we can enhance this color using this compound here, which is called orthophenolphthalein, which is colorless itself. But when it complexes with iron, you get the really nice kind of orangey color. And this orangey color can be measured by the instrument. So this is what you're doing in this week's lab. We're getting some samples, and we're going to measure iron in them. We have to add this compound. I can't remember, there's a few other things we have to add. I believe that this compound is sensitive to acid pH reactions. So I can't remember, I think we add a little bit of base just to make sure that the thing doesn't react with any acids in the water and so on. But this is the basis behind this week's lab. So how does this work? If we don't know the exact relationship, we have to make something called a standard curve or a calibration curve. And so this is going to be kind of part A of this week's experiment, is that what you're going to do is you're going to make up some known concentrations, right? So you're going to make up a 0 concentration of iron solution, and I can't remember the other concentrations. It's like 5 and 10 and 15 ppm or something like that. And so when you have your known concentrations, right, you can measure your absorbance, and then you're going to make this thing called a calibration curve. So this calibration curve is just a direct linear relationship, and this tells us exactly how much these things are related. So you probably know with a curve like this, we can determine the equation of the line. So you remember y equals mx plus b. So b is the intercept, and if your intercept is 0, 0, then we don't need to worry about b. So what you're doing is just figuring out your slope. So your slope, of course, is going to be your rise over run. And then we can use this y equals mx formula to determine an unknown, right? So here's an example. It says, we have an unknown, and the unknown absorbance is 0.25. So there's two ways you can do it. One is you can basically just find it on the graph, that 0.25, and then you can get a ruler and draw down and see that it measures to 5 ppm. The other way you can do it is actually, like I said, using the formula, which I meant to put on the slide, but in this case here, the formula, y equals mx. So y, of course, is absorbance, and the slope, I haven't calculated it, but the slope is rise over run, so it's about 0.5. It looks like 0.5. Rise is 2. Rise is 1, yeah. The slope is about 0.5, which I haven't calculated there, but and x, of course, is the concentration. So in this particular pigment, the concentration, of course, is going to be 0.5 divided by the absorbance. So like I said, two different ways to do it, and that's what you're going to do for basically this week's lab. We're going to do 3 samples in the lab. I think, if I remember correctly, we have tap water. We always like to do tap water. I think we're doing the groundwater, and then I have a sample of stream water that's known to be high in iron. So that's kind of it for spectroscopy that I wanted to say, by the way. Like I said, you're going to get experience using the instrument and doing these calculations in the lab. And it's a pretty standard thing to do, is to figure out the relationship between these things to make a calibration curve. Because like I said, every pigment and every solvent's a little bit different. So often these calibration curves have to be done in order to figure out exactly that relationship between the absorbance and the concentration. So I think I mentioned last day, I asked if you had done the colorimetry in the lab. We have a little instrument. It's a little handheld instrument. It kind of looks like that one shown in the diagram. By handheld, it's bigger than a cell phone. But it fits in your hand, and it's a little meter. And colorimetry is kind of just a really simple, basic form of spectroscopy. So you have one instrument, and that instrument is not even separating out the wavelengths of light often, but it's calibrated to measure a certain wavelength and calibrated for one particular type of solute. So the one we have down in the lab, maybe I'll bring it to lecture sometime and give a demo, is used to measure fluoride in water. And so in that case, what you do is this kit is set up something like this. You get a vial and you add your water to the vial, and then I think you add something like 5 drops of a certain solution, and it turns sort of a pinkish color. Then you pop it into the instrument, and the instrument gives you a number. It's already programmed to convert everything to parts per million. And so colorimetry is kind of like spectroscopy, but like I said, it's not as expensive or sophisticated in terms of measuring specific wavelengths. Is usually not necessarily done. Kind of depends on the instrument, but it's pretty common nowadays. I know when I went to one of the, I'm trying to remember which water treatment plant I went to, they had a colorimeter, and it was programmable for the lab technicians, so they could select what it was they're measuring. So I think they had phosphates and ammonia, and I'm trying to remember the third thing that they were measuring. And basically, it was already preset. So they didn't need to worry about wavelength or anything like that. They just had to make sure the right reagent was in the vial, and it would just directly output a number. So colorimetry, like I said, is kind of like spectroscopy. It's a little easier, a little cheaper, and it's mostly for convenience. So you're looking at these kits dedicated to a certain compound. Okay, so last part of today's unit, I want to talk about field sampling of water. And this is one of these things that I have a little bit of experience doing this, but it was a long time ago, like literally when I was a high school student. I had a summer job doing some of this, and that was the last time I've done it. But the reality is when we sample things, there's going to be a lot of instructions given to you by the employer or the lab, and these are some of the points that I want to emphasize. So why do we sample water? I think that one's probably obvious. There's a lot of reasons why we're doing this. Some of this is routine. It's mandated that we sample water, for example, in areas where, around landfills, around, industrial operations. It's essential that water gets measured to make sure we're not contaminating the environment. So a lot of this is routine. Sometimes there are investigations where people are concerned, and then they, are going to certainly check things. And of course, we've got research and all sorts of other variety of reasons. So the goal, of course, of sampling is you want to get a sample that is representative of the source. So what I mean by that is that, you go and you arrive at a pristine river, and you're not sampling the water in a corner where there's a bunch of sludge or someone has, dumped out their lunch, right? You want something representative of that river. So that's part of the goal behind sampling, is that you are getting something that is representative of the water body in question, not that one sample that's near a dead fish or something like that. So something to know about sampling is understanding the procedures. And like I said, procedures will be different for different companies and for different purposes. And also even it's important that you do talk to the lab because depending on what kind of analysis they have, they may have different requirements in terms of how you collect your sample, how you store your sample, and those kind of things. And I'll give you some examples in a minute here. You can see some kind of common questions. How much water do we need? Do we need 50 milliliters? Do we need 2 liters? Those kind of things, right? And so the lab usually can answer those kind of questions in terms of, is it going to be kept in a plastic bottle? Is it going to be kept in a brown glass bottle? Those kind of things, depending on the actual test that's actually being done.
So what's usually like when you're out in the field, like a field sampling? What's a common thing?
A common thing
I don't know
are
if
these,
you to test.
A common thing are these plastic bottles, right? Some tests are done out in the field. So pretty typical tests that are done out in the field are things like temperature, dissolved oxygen, pH, and conductivity.
Have you ever had to, in your personal experience, have you had to go out in the field to Blaine? What have you ever tested if you did?
Well, so when I was a high school student, I worked for 2 summers with a biologist, and we did some lake surveys. So a lake survey is Basically, we went out into the middle of the forest to these lakes that had never really been studied, and did a bunch of basic studies around the lakes. So we caught some fish and saw what was living in there. We tested the pH of the water and conductivity. We measured the depth of the lake. So it was really just very basic stuff that I had done. Like I said, it really depends on what the whole nature of the job is that people are trying to do. And pretty standard for people sometimes to, like I said, collect some samples in these plastic bottles. And, you know, sometimes there's questions in terms of, you know, what analysis we're looking for. Like I said, with field meters, you can do pH and conductivity very easily, and temperature and dissolved oxygen. More sophisticated tests, often they're sent away. So one thing they've done is just to keep things fresh, just like food, usually the samples are put on ice. And so people will bring out coolers and you know, put the bottles on the ice just to make sure things are kept fresh. So I have some examples of specific things that could be measured, and this is actually this was actually a presentation I went to from Maxim Labs. They do a lot of water chemistry tests, and so these are basically their guidelines from their labs. And so you can see here's the plastic standard bottle, and the common parameters, you know, the whole bunch of parameters are measured using these plastic bottles, right? So you can see the usual ones: pH, conductivity, color, phosphates, sulfates. Most of these things are not things that degrade very quickly. They're not light sensitive or anything like that. They're pretty standard kind of tests, and so they can be kept in a bottle. Their preference is that you keep them cold. But depending on the test, that may not even be necessary. Just keep in mind, in the field, the water itself is usually not too warm, right? Sometimes looking at certain nutrients like ammonia and those kind of things, these things can break down, and so they actually have these special bottles that have a little bit of sulfuric acid in them. And sulfuric acid is basically preserving, keeping the whole thing acidic to preserve these compounds. Yeah.
I think you made another typo. Ammonia is NH3, not an NH4.
Oh, there it is again. Hey, yeah. So, yeah. Although in water, it is often ammonium with the positive charge on it. So, let's see here. Maybe I will just fix that right now, because you know what, I will get back to my office and I will totally forget. I suppose that disabled the sharing for you guys. Just give me a second here. I'm going to fix that because, like I said, otherwise I will forget. Okay, thank you, Mr. Eagle Eye. I do appreciate it. I'm not making fun of you. Okay.
Oh,
Yeah, so like I said, there's lots of different things that people are measuring, and these are just some examples from Maxim. So you can see some of these less common parameters. So these metals. So we've got, what have we got here? So if you remember your periodic table, Ag. You guys must know that one. Who knows what Ag is? Yeah, Ag is,
it's silver.
Yeah, that's the big money, right? That's the one we want, isn't it? Aluminum, arsenic, so all these things here, oh yeah, there's another typo. I have a superscript instead of a subscript for HNO3. But yeah, these things, they must degrade or something like that. And so this lab has determined that this is a good way to preserve it, is by having a little bit of nitric acid in that particular container. You can see there's other special things that sometimes people are looking for sometimes. So mercury is clear glass. So I don't know in terms of the chemistry, but maybe mercury reacts with plastic. Some organics are photosensitive, and so they put them in the brown glass, and that's supposed to protect it from stray light. So you can see, for example, naphthenic acids and pesticides are an example there. And volatile, and there's our microbiology coliform jars as well that have some preservatives as well. So like I said, there's a whole bunch of these. When I was looking at the Maxim list, I bet you there must have been at least 30 different types of jars of different sizes and bottles of glass and plastic for measuring all sorts of things. And you can imagine that there's quite a number of different things people are looking at. So what kind of things are people doing to prepare? Like I said, you know, the background information when people go out in the field is important, talking to the lab and figuring out, you know, when you're going to sample a place, right? You know, is this public land, private land? You know, how to get in there, all those kind of things are obviously important. Sometimes permits are required or permission is required. We had one of my former colleagues here was doing some water testing, looking at naphthenic acids. And so she was going on Syncrude land. And in that case, of course, she had to go through and jump through a bunch of hoops and get permission and all those kind of things. They wanted to know exactly what she was doing and whatnot, and before she got the permission. You know, there's usual safety precautions, you know, I know she had to wear, I think, because she was near the tailings pond, she had to wear a life jacket, but, you know, a lot of places will require you to wear, you know, the typical safety stuff, so steel-toed boots and hats and those kind of things. And of course, gloves are important, right? And I'll talk about gloves here in a moment. And you can see this last part, correct documentation, because some things have to be documented at the field, some during and some after. Right? And I think I have a form somewhere here to show you what it might look like. So a common procedure for water sampling is this thing called dirty hands, clean hands. And I have a video to show you this in action, but the idea is here is that you have one person in the partnership is doing the dirty hand stuff. So what does that mean? It means that they're you know, handling the documentation and you know, touching things that could get into your water sample, opening outer Ziploc bags and operation of pumps and those kind of things. And the clean hand person is the person that is directly handling the container, sampling the water, and putting it in the bag. So I'll show you an example here. I found a nice video demonstrating this on YouTube. And so I will play this for you. Let me just load this up. And I'll have to share it once it gets started.
Hi.
Okay. And these two ladies are, I'm not exactly sure what they're sampling, somewhere out in the snow. I don't know if it says down here at the bottom. Let me see here. They're sampling mercury near Winnipeg, or it sounds like. And anyway, they're going to demonstrate the clean hands, dirty hands technique. And this is a pretty common thing that's expected of a lot of field sampling in order to minimize contamination of the samples. So I'll play that for you right now, and hopefully you'll get the idea from that.
I'm Amanda.
And I'm
Why do we do clean hands, dirty hand sampling?
Well, Sarah, when we're sampling for mercury and other trace contaminants, the biggest source of contamination is humans. And we want to make sure that we're not adding any contamination to our sample and we're getting an accurate measure of what's in the environment. Specifically for mercury, we're actually looking at the parts per trillion level, so we have to be very clean when sampling.
So remember, don't be afraid of your sample. Your sample's afraid of you.
That's right. All right, Sarah, shall we start?
All right, so I'm going to be taking on the role of dirty hands today.
Haha.
That means that my job is to physically reach into the cooler, put gloves on myself, gloves on Amanda, and then touch all of the different sampling bags.
Except, as clean hands, my job will be to touch the sample and the falcon tube, which is inside the bag, which you'll see in a second. Shall we begin, Sarah?
Yes, Amanda, let us.
All right.
Note that the gloves are all put in separate bags.
So Sarah's going to start and put gloves on herself. And because I'm clean, I cannot put gloves on myself because I will contaminate them.
Remember, Amanda only touches the sample bottles.
Nice glovemanship. It's kind of like being a surgeon.
It's true. Except it's colder here. Are you ready, Amanda?
I'm ready, Sarah.
Let's go around.
There's a lot of activity this morning here at Surf.
Now, Amanda's already doing something she shouldn't be, and she's talking. When you're doing your sampling, make sure, as dirty hands, your clean hands person is not talking. You can yell at them, you can point at them, just make sure they're not talking as much when the sampling's going on.
And why shouldn't I talk, Sarah?
You shouldn't talk because even breathing on the samples can introduce a source of contamination.
Especially if you're a smoker, which is super gross.
All right, now that we are fully gloved up, we are going to start taking our water samples. Again, my water samples are put into individual bags. As dirty hands, I can remember to touch the bags. And we are first going to start off with taking the samples. I have my triplicate bottles here.
So again, I'm not doing anything because I'm clean.
And now, as dirty hands, it's my job to fiddle with the tubes.
But not touch them.
I push them up, don't actually touch them, and I pass them over. Second sample.
So we repeat this process 3 times to collect all 3 samples.
Okay, I'm going to stop it there, but I think you get the idea behind clean hands and dirty hands. So the whole idea, like I said, is to make sure that the person who's actually touching the bottles and touching the samples and all that is as clean as humanly possible to minimize contamination. Like she pointed out in her case, they were looking at parts per, what did they say, parts per trillion? Yeah, that's like not a lot. So you do want to be very, very careful when you do those kind of things. By the way, that's a pretty common environmental kind of job interview question, you know, to explain this kind of concept, particularly if you've had some field experience. And so just a tip there. I know I've talked to a couple people that have said they have had that on actual interviews. So understand the principle behind dirty hands, clean hands. So I think I talked about some of this stuff here already. Let's see here, just going through that list. Like I said, you know, talking to the lab is really important because sometimes they're actually asking you to fill the bottle completely, so you'd be filling it under the water and putting the cap on under the water. Some tests actually want a little bit of air in the top of the tube or the bottle as well. So something, like I said, you know, that's going to be specific for the test you're doing and what the lab is going to desire and so on. So all these things, you know, are the types of things you'd be learning on the job because they'd be, you know, telling you to do it, but just some tips there of things that you might see. So the last thing I want to talk about was paperwork, right? And again, depending on the kind of paperwork that is done, you know, sometimes samples are taken for legal reasons, right? So maybe there's an investigation going on, or the workplace wants to, you know, make sure that everything is done properly and that, you know, if there's any questions. So sometimes, you know, sampling has to be witnessed. There has to be 2 people signing the paperwork and all those kind of things. And so, of course, that's something as well that has to be carefully done depending on what it is you're trying to do. And I'll talk about chain of custody in a moment here. So I think we had talked about this already. These kind of tests are often done on site. So dissolved oxygen, pH, temperature, conductivity. Usually there's field notes, and the field notes would include, you know, things like your coordinates and those kind of things. You can see it's mentioning in the notes the chain of custody, which is something I want to talk about for a minute. So chain of custody is really important for legal samples. Not necessarily all samples. It may be required by your workplace, but any legal sample, so like I said, an investigation or something that's part of a record that must be legally kept by a company requires chain of custody type of paperwork. And so I want to talk to you about what that means. And here's kind of a sample of a chain of custody form. And so the whole idea is to follow the sample along the entire route. So who collected it and when, and as soon as that sample is transferred to somebody else, the chain of custody form is kept track. So let's say you collected the field sample and you deliver it directly to the lab. The moment when it arrives at the lab is going to document that on that piece of paper to show that it went from you directly to the lab. In some cases, it doesn't happen that way. Maybe you're doing the field work and someone is coming and collecting the samples at lunchtime, And so that chain of custody form will be filled out at each time it changes hands to make sure that, you know, nobody's, it's not getting intercepted or anything like that. And I know it sounds really silly, but there have been a number of cases where these things, the evidence has been inadmissible in court because chain of custody wasn't paid attention to. Here's an example, not of water samples, but this is a drug test. I'm not familiar with baseball or this particular player, but he was suspended over a positive drug test. And the judge overthrew the suspension because of chain of custody issues. So you can see what happened here was that the sampler went home after collecting the sample and put it in his basement fridge. I think the story was that the lab was closed by the time he got there, but this meant that the sample had left his hands and went into his fridge. And so I think the protocol in that case, I'm not entirely sure. I think he would have had to document it that it was stored in his fridge and somehow demonstrated that his fridge was locked or secured or something like that, because of course, you know, it was possible someone could have contaminated with it. And so no evidence of tampering, but the judge had ruled that the chain of custody was broken and that the evidence was inadmissible. And so he got off scot-free for doing whatever drugs he was doing. I think I mentioned this already, you know, storage, you know, again, you know, this is usually on ice and sometimes bubble wrap and things like that to protect glass containers. So there's one more picture for you, and you can see with these legal samples, often, so Maxim has a system at their lab for dropping off samples after hours. So it's pretty common for people to do their field work and they don't get to town until, you know, 6 p.m. or something like that. And I think Maxim closes at 5. And so they have a system where you can drop off a cooler and you lock the cooler into some sort of locker, and there's these chain of custody stickers that are apparently tamper-proof somehow. So you must sign your name over it or something like that. And that's how one way that they maintain their chain of custody for those kind of things, which is pretty common, right? Like I said, people arrive from the field a little bit late, and that's the situation. Okay, so I don't know if anybody has any questions about that. Like I said, a lot of this stuff is going to be job specific. You know, whatever you're sampling, it's specific for that. But these are some of the things you'll learn about when you do go into the workplace, particularly if you're doing field work.
Blaine?
Yep.
Will any of these jobs require like you to go like diving or something? Like you have to wear like a diving suit or something?
You know, that's a good question. I have not heard of that. But I can tell you when I did the lake surveys, there are methods for sampling water in a lake at different depths. There's the system we used, I can't exactly remember how it worked, but it was like a cable, and there was a weight on the end, and you would sink it down, and on the cable were measurements. So you could say it's down, you know, let's say 5 meters of depth or 10 meters of depth or whatever. And then there was a little pull cord that went through the center of the cable that would basically open up a little sampling container on the end, and then you could close it, and then you could pull it up and deliver that to your bottle.
Because there are like some areas of like the Clearwater River where there's like at least 50 feet deep, so. Yeah,
and there's methods for doing that without diving, but who knows? Maybe if you get lucky, you get to do that. I did do some scuba diving years ago. It was a lot of fun. Okay, so we have a few minutes left, and I am going to introduce the next topic. I just need to switch out of this PowerPoint. And okay, what's going on here? Find my other one. So topic 5, at least what we're going to cover today, is going to be a bit of rehashing of some things we talked about already. And so let's see here, share screen. So topic 5 is hydraulics and hydrology. We've talked a little bit about hydrology already, which is basically water and how it moves around the environment, right? So, you know, we've talked about the water cycle in particular. So we just want to kind of recap a few things along that lines. And next day, we're going to talk about hydraulics. And hydraulics is also movement of water. And usually by hydraulics, we mean water through pipes and things like that. So we're going to cover that next day. And these are all part of us, of course, you know, when we extract water from the environment, all these things are very important. So I think I showed you this exact slide before. Hopefully you can remember all the 6 processes of the water cycle. We had evaporation, condensation, precipitation, runoff, infiltration, and transpiration. And all of these things are part of you know, the earth sciences. And you know, people study these things in terms of, you know, how fast groundwater moves and things like that. We're not going to get into these in a lot of detail. Like I said, I'm mostly just recapping some of these concepts. And you know, the big one that I find when we're talking about the water cycle, the 2 that people usually forget are surface runoff and infiltration, and a lot of people will forget transpiration as well. So hopefully you remember what transpiration is. Anyone remember what transpiration is?
Evaporation of water from plants.
Yeah, so it's kind of like plant sweating. And I think the numbers are that that's probably about 10 or more % of the water in the atmosphere is just from plants sucking it out of the soil and it basically leaving through their leaves. So it is quite significant. And that's why in jungle, you know, rainforest regions, you know, that's contributing to the local climate in terms of the, you know, daily or whatever rain that occurs, because the plants are just sucking so much moisture out and putting it into the air. So like I said, what we want to talk about and what we're getting to is kind of talking about human use of water. And so another way to think about the water cycle is to think about the urban or human hydrological cycle. And so you can see in this case, we have water being taken from a source. Usually before we use it, we treat it. And so, you know, we have a drinking water plant. It gets distributed to the people, so through pipes or trucks or something like that. And then before it gets returned to the environment, usually we're treating it in some form or another. And so that would be a wastewater treatment plant. And like I said, these things we're going to talk about in a couple of units from now in quite a bit of detail. So some other reminders of definitions. I can't remember if I gave you this one before, but limnology is a study of inland waters. So this can include all sorts of things, salt and fresh water, lakes, streams, rivers, wetlands, and those kind of things. And often when people talk about limnology, they're kind of pairing that with the ecology of the system, right? Looking at the wildlife there and plants and all those kind of things. We talked about as well, I think back in topic 1, I threw these definitions at you, lentic and lotic systems. And these are describing freshwater systems that are either still or flowing. So lentic is still, so you know, lakes and ponds and marshes, and lotic is flowing, so rivers and creeks and streams and those kind of things.
Blaine, I have a question.
Yes, certainly.
Would the ocean wouldn't be considered lentic or lotic, would it?
No, not usually. Usually we're talking freshwater, lentic and lotic are freshwater systems, right? And limnology is inland, so meaning not seas and oceans usually, yeah. I mean, sometimes the words are a little funny because sometimes when we say seas, like if you know about, you know, the Sea of Galilee, which is found in the Middle East, is actually a lake. It's a freshwater lake. And it's just the name stuck, right? It's a big, you know, a big lake inland, and for whatever reason, people call it a sea. And then you've got Salt Lake, which is in Utah, and that's an inland saline water body, of course, right?
Yeah, and have you heard of the Dead Sea?
Yes, of course. Yeah, you always see the pictures of tourists that are floating there because it's really, really high salt, and so you apparently float really easily, right?
Yeah, and I heard nothing can live in there.
Yeah, that's a good question. I've always wondered that, whether there's any ecology there or not. There might be some microorganisms, but that's just a guess. I think that's an area that's supposed to be getting saltier too, because it has no outflow, right? And so it's only inflow, and it just collects salts over the eons. So lakes, of course, are important in Canada, particularly as a water source in many areas. And by lakes, I often mean reservoirs too, as well, because a lot of reservoirs are basically human-made lakes, and not as common that I know of in Alberta. There are a few, but you know, in southern Ontario, where I lived for about 10 or 15 years, it's pretty common. A lot of cities will have man-made lakes that are basically reservoirs for drinking sources. Here's Rainy Lake. This is a place I visit every year. I thought I'd show you a nice picture of that. So things that are measured on lakes, you can see there's a whole bunch of things there, right? Surface area, depth, color, turbidity, all those kind of things are often important parameters that people are looking at for drinking water. And of course, understanding how healthy it is for wildlife. Obviously, the formation and history of the lake is going to be important. This is Abraham Lake. I can't remember if I mentioned this one before. This is west of Rocky Mountain House, so kind of halfway down the province. And I guess I was hoping that picture showed it a bit better. Abraham Lake is actually a human-made reservoir. It's very beautiful if you ever get the chance to go there. It has that sort of really aqua green, kind of bluish color to it, and that's because it's got all that silt from the mountains going in there, and that reflects the light in a unique way. But also makes the water cloudier and less more particulates in there for drinking purposes. So some things to consider about lakes. I think I mentioned some of these things already, how it's formed, size and shape, human activities, all those things are pretty obvious things. So another reminder of things that I had talked about before is that lakes can have, in places like Canada, they have overturns in the spring and the fall. So an overturn is kind of just when the water is changing temperature. So if you remember that water is most dense at 4 degrees. So when water gets colder than 4 degrees, it gets less dense. So some of that colder water actually moves to the top. And when things warm up, we've got things moving to the bottom. So this is fall and spring overturn. And a lot of these things will actually stir up nutrients, change the dissolved oxygen levels, and all those kind of things that are important for water quality. There's some other terms for you. Epilimion, I can never say that word, so I'm not even going to try again. We also talked about aquifers, of course, as water sources and water flowing through these. Do you guys talk about groundwater flow in Neil's classes a little bit? Yeah, it's not something I know a lot about other than it's slow, very slow. And so some terms, again, I've been trying to reorganize this course over the last couple of years in terms of some of this material end up earlier, and I apologize if I'm just repeating myself. But this line here, right there, is the water table, and that's where the unsaturated zone and the saturated zone exists. And so this is something that's really important for people drilling wells because they want to go beyond the water table. And if the water table varies over the seasons, they want to make sure they go deep enough to make sure they're always below the water table, at least in theory. So what do we use as aquifers? So anything that's porous and can hold water. So sand, rocks with pores in them, and these are some of the common types of aquifers in there that you can see in terms of the geology. Okay, so sorry, I'm just last slide here, and we'll come back to this as well. And how do you make a well? You drill a hole, you hopefully get beyond the water table. And usually there's a screen or a filter or something down there that's going to take out any particulates, and that's where you're going to have some sort of pump in there. Anyway, sorry for going over time. We'll come back to this next day. We're going to talk, like I said, we're going to do a little bit of math next day, talking about water flow and pressures and things like that. Formulas are pretty easy. The hardest part is doing the metric conversions. So good luck with tomorrow's lab, and I will see everybody next week, and have a great weekend in the meantime.
Thank you.
Have a good day.